Pretty thighs, shawty, she a dime. Demon girl, evil eyes, she be telling lies. Slim waist,
So today it is a pleasure to interview Dr. Schlessinger, a professor at Yale and an accomplished entrepreneur. He has founded a bunch of biotech companies like SUGEN and Plexxikon. So, Dr. Schlessinger, thank you for being on this episode. Can we start with you taking us back and telling us about your background?
So I was born few months before World War II ended in what used to be Yugoslavia. My family is Jewish on both sides, and we lost almost all my family. I was born under very sad circumstances. My parents escaped, and they were captured by the Italian, who was an ally with Germany. And they were put in some kind of concentration camp on an island. And when Italy, each of them has its own first family. So they were both widowers. My father also lost his daughter, half sister of mine. And so when Italy surrendered, they rebelled, and that they joined what they call the Partisans of Tito. This is a communist army that was liberating. And for the last 2 years, they were there. And I was born there at the very last 2 months of the war. Immediately after the last just before the last German offensive. And so then we settled up after the war, and my father, my mother, and my grandmother in Yugoslavia. And then because of the harsh situation of the communism there, my parents decided to emigrate to Israel. So in December 48, we immigrated to Israel because one of my uncles and aunts were already there in a kibbutz. So we settled there for a few months, and then we moved to a small town, and this is where I grew up in Israel, not far from the city of Haifa. And so I did all my school there, all my education, and so sort of do you need more details about it, or should I proceed with this?
I think it's good. I mean, I want to ask something specific, but
yeah.
Okay.
You can always interrupt me and ask me.
Yeah, okay, yeah. So the next question is, I think in your bio, you said you served in the Israeli army in the military. So by the way, this is the first time we've had a veteran that fought on the front lines actually in the war. We had a previous one, but he was in the Israeli intelligence, so it wasn't in the front lines, but in the backstage doing all the work. So I'm going to ask you, what was it like serving in the army, and what was your role in the army?
So I was eventually a captain.
Oh,
yes, I was a.
You have a medal? You have a medal?
Well, but these are all I participated in actually 4 wars, 4 battles, short battles. And I did not like the idea of military, but since it was compulsory, I had to go and did it. And then, of course, very quickly in my life, I realized that you can control your destiny in a better way if you are involved in the decision making. So this is why I decided to become an officer. And I was basically what they call a SAPER. These are Rangers unit, which are expert on explosives. And I was part of a Ranger brigade called Golani Brigade, which was a very famous, and we occupied the Golan Heights. So I participated in quite bad battles several times in my life, and this really had an important mark on my life in many ways. You would be amazed to what extent, when you are 22 years old and you take 50 people to battle, to what extent it shapes your life. You mature in no time. And, people try to teach here in the universities about leadership. I mean, you have to develop leadership instantaneously when you go to war with 50 people, And the key aspect which I learned during my early life about leadership is to respect everyone that is under you. And you have to treat everyone as equal. And I have really used this throughout my entire career to realize. So I participated in the 6 Days War, in the Yom Kippur War, and in some attrition war in between, and the last one was in 82. And I spent 2.5 years in the military and then about 8 months in reserve during all those years.
Yeah, I mean, and the question is, while you were clearing minefields, did you ever scare you were going to step in one, or do you have any like near-death experiences that you would possibly share with us?
Yeah, well, look, in a way, I wouldn't say I enjoyed this though, but it was, if you deal with explosive and mines, it's more scientific than other fields, but you're also part of the infantry and tank unit. So it's sort of, it's really telling you a lot about priorities in life. And if you're really close to death, and there were few opportunities when I was under serious fire, it really changed a lot. And it was particularly tragic because my parents, it was a tremendous burden of my parents, they lost everyone. Here I am in a war. So it really shaped a lot of my pace. I did my degree in chemistry and physics, and I was thinking more about being a physicist. And then I sort of realized at a certain stage that chemical sciences and biological sciences are going through a revolution, and it should be part of it. And when I actually finished my PhD degree at the Weizmann place called the Weizmann Institute, I was planning, ironically, to apply for a position of postdoctoral position at Yale University. This is now in 1973. And amazing, and then I went to this war, and for 6 months, and then I came back, and I had to refresh myself and get back into normality. And then there was some kind of meeting in which a lot of international scientists came to visit Israel. This was in early 74, when I met someone called Elliot Elson, who is a professor at Cornell, and he proposed that I joined his lab for postdoctoral training. So this was, everything was delayed. So I arrived to Cornell University in January 75 to work in the lab of Elliot Elson and Watt Webb. And they developed this method called fluorescence correlation spectroscopy and fluorescent bleaching experiments. So I was a pioneer in that field, doing these experiments, studying how molecules move on the cell membranes. And I became interested at a very early stage in receptors. And, but at a certain stage, I realized that biophysical measurements are not sufficient to understand how things work. And I was reading the literature, and I found that in the literature that certain cancer cells, in order to grow better, they produce some of their own growth factors. So I became interested in the field, and so I was surrounded by excellent physicists and chemists at Cornell University's physics and chemistry department, but no one knew anything about cell culture. So I decided to go and take a course at Cold Spring Harbor Laboratory to learn about how to grow cells, just because no one knew how to do it, and someone. So I learned that, and there I met someone called Graham Carpenter, who was working with Stanley Cohen, who discovered EGF, epidermal growth factor, and he gave me a small amount of EGF to do some experiments. So this is how I became interested in studying potential receptors and growth factor, which turned out to be a very, very good decision. And there is quite a lot of literature already that certain cancer cells produce their own growth factors.
Yeah, I mean, I want to go back to, I guess, post-military when you were in university. Did you have any PTSD issues after you came back from your wars? And like, how did it affect your family? Because I think especially right now, PTSD is a huge issue for a lot of former Army people coming back from the war, where it is in Iraq or something. So I just want to ask your experience.
So this was not so much discussed at that time. I clearly had some issues.
Mhm.
But, you know, we were different at that time. We did not complain so much unless it was totally paralyzing. I had another issue, which was at the end of my military service, as in the first few months at my college education, my girlfriend was killed in a car accident.
Oh,
And in that car accident, while in the military, there were other 4 people that I knew. And so I was very depressed because of that.
mm.
Then, of course, to some extent, ironically, when I went 2 years later to the 6 Days War, the horrors of the war somehow put a shadow on my private depression. So I would not know how to separate between my losing my personal having my personal loss and the public loss which I was involved, getting to treat people in the battlefield. And so I was clearly very much stressed. I don't know how to define it, but what was really amazing, as soon as I arrived to the US and landed in this place, Cornell University in Ithaca, which was surrounded entirely by science, I felt that I came to an intellectual heaven, and I became very relaxed, and somehow it's like another life. It was like another life. And this was kind of amazing. So I do not know exactly how to answer my question, but there was a major impact on my mood and focus. And the only way in which I dealt with it is just thinking about future and hoping that the future will be better, and which turned out to be the case.
Mhm. Well, that makes sense. You think something positive to
I
get
would
rid
say
of the
I
yeah.
developed this attitude of positive thinking.
Yes, that's really good advice, because I watched a documentary on Chris Kyle. You probably know who he is. He's an American sniper, and he has some PTSD issues. So I was asking your experience, especially.
I saw people with severe PTSD in the battle, people sitting with shock on their bodies of their friends and not even noticing that they're sitting on a person.
Wow.
Yeah, you see that, and I have been involved in some terrible accidents of explosions,
Mm-hmm.
which you find a way how to deny it and somehow focus on the task, because if you're really so much controlled by anxiety, you cannot function.
Mm-hmm. Interesting. Okay, and I know you mentioned how your military experience impacted your scientific career, but how about your business career? Like, how did the stuff you learned during your time in the military impact your future
Well,
in the biotech
I
industry?
think when you are subject to extreme issues of the type that I experienced when I was 22, going for the first war, it really taught you a fantastic lesson of how to deal with people. And as I told you before, all of us are equal. We are treated with respect, and I think this served me throughout my entire career. And then, of course, I thought that I had leadership, because you have to make decisions.
Mm-hmm.
In the second war that I took part, which was Yom Kippur War in 1973, I was already a father. I was leading like 80 people, and I wasn't a captain. And so you know how to organize, how to make plans, how to deal with personality. All those things serve you very well in running a lab, and of course in being involved as a chairman of a board of companies, which I did for several times.
Mm-hmm. So we're gonna move on to your scientific work. I know your lab cloned a fibroblast growth factor receptor. So my question is, who thought of this project, and how hard was the cloning process?
So FGF receptor is one example. So my lab was involved in studying these molecules, receptor tyrosine kinases, before they were known to be tyrosine kinases. So we started to work on it when I moved to NIH. After doing a postdoctoral training at Cornell University, I decided that I need to increase my knowledge in biological science. I was quite good in physics and chemistry and biophysics. So I took another position at the immunology lab at National Cancer Institute with someone called Pierre Hankart. And then I established a collaboration with people in Ira Paston lab at the Cancer Institute. And we started to study receptors. And so one of the interesting things which I discovered at that time is that there are certain antibodies against receptors, which insulin receptor, which mimic the response, which was kind of very interesting because at that time it was not known if like EGF or FGF or any of insulins enter the cells and induce a response, or they activate the receptors. And the fact that an antibody against insulin receptor, which was isolated from patients, can stimulate the response, indicated that the information is in the receptor. And the function of the hormone is to activate the receptors. So I became at that time very much interested in purifying receptor and cloning them. And in fact, the first receptor that we were purified was EGF receptor, much before the FGF was cloned and all that. And I did it at the Weizmann Institute when I was a professor there. After postdoc, I became a professor at the Weizmann Institute. And this was at a very early stage of cloning. And there were very few labs at that time that knew how to obtain microsequences. In order to clone at that time, you had to isolate the protein, prepare a few peptides, and then use it to develop to screen cDNA libraries. So I collaborated with someone called Mike Waterfield, who was a very good sequencer. And this is when we made the first important discovery, when we together his lab, Julian Downward, a student in his lab, collaborated with a student in my lab called Yossi Yarden, obtained few peptides from EGF receptor. We realized that these peptides are identical to a viral oncogene. So this was the first link between a receptor tyrosine kinase and oncogene, and shows you that cancer can be caused by aberrant activation of a normal receptor tyrosine kinase. So this was very important. And at that time, there were very few people who do cloning in a robust way. So we collaborated with Axel Ulrich, who was a genetic at that time. And we published a series of papers in 84, 85, in which we opened this field, where we showed that EGF receptor is an oncogenic protein. My lab showed that it's amplified, the gene is amplified in multiple cancers. And then, of course, we also showed that we have antibodies, that some of them mimic the response of EGF, which again confirmed the idea that the information resides in the receptors, and the function of EGF is basically to stimulate the activity. We came up, once we looked on the topology of how this receptor look, and there are molecules which have a single extracellular ligand binding domain, a single transmembrane domain, and a cytoplasmic domain with tyrosine kinase activity. I came up with the idea that the function of the ligand is to induce receptor dimerization, because this way you don't have to transfer a signal through the plasma membrane by pushing and pulling it, but rather by creating dimers. And this was all happening in the mid-80s.
Mm-hmm. So my next question now is about your companies. You've co-founded a lot of companies like SUGEN and Plexxikon. So who thought the idea to found SUGEN?
Okay, so this is an interesting story, but I have to give you a significant background about it. So it all stems from my research. So one of my collaborators was Axel Ulrich. So Mike Waterfield died 2 years ago, but Axel is retired. He was one of the early scientists in Genentech. And once we worked out EGF receptor, I developed a strong scientific relationship with him and really became very close to Genentech. And this was a very early success. And basically, Axel Ulrich, through him, I sort of got to know what it takes to develop drugs in a company. And so, but I was not planning to do anything, and I had no clue about it. But at certain stage, there was a time for me to get a sabbatical leave from the Weizmann Institute. So one of the fellow who was my boss at NIH became a director or president of a company at the Washington area called Malloy Lab that became part of Royal Pharmaceutical at certain stage. And he suggested that I move and take a sabbatical in a company just for 1 year. And then I told him, well, it's just for 1 year. So I said, well, you never know, maybe you can stay longer. And so I took this, and this was in 1985-6, and they offered me fantastic conditions and fantastic terms. I had a large lab there, and I could do whatever I wanted. So I was really employed by the company, and they sort of asked me to be head of a department. So I did not quit for my job at the Weizmann. So I would spend like 2 months in my lab in the US and 2 weeks in my lab in Israel, and I did it for several years.
That's a lot of flying around.
Yeah, but flying back and forth and all that. And I was prepared to do it for a while because I did not want to quit for my academic science. And I was in my 30s, I couldn't do it when I was much older. And at certain stage, this company was sold, and I had some shares, and suddenly there was tremendous values to my shares, and which actually changed my life. So suddenly I found myself to be a part of a company, and I came to the US without any intention to stay nor to make money, just to do science and spend. And so it became clear to me that this is very interesting. And that, of course, this is quite lucrative. So I started to think very early stage about developing inhibitors for tyrosine kinase.
Mm-hmm.
Okay? So I was proposing it to the company, and in fact, they were really starting this program, but there was a lot of resistance about blocking the activity of kinases with ATP analog that they might be toxic. So there were several reasons why I did not want to stay and be employed in the company, because I thought that the freedom that academia allows you and the fact that you can really work on things at your own decision are very important. So after 4-5 years there, I decided to go back to academia. And I was considering to do it at the Weizmann Institute, but there were some issues there that prevented me. So I eventually took a job at NYU Medical Center as the head of the pharmacology department. And how is it linked to started companies? Because Axel Ulrich, who was working for many years at Genentech, decided to take a job in Germany as a director of a Max Planck Institute, which are very good position at the in the German science. And then we realized that we have quite accumulated knowledge in what it takes to develop drugs, So we decided to start a biotech company. So this was in 1990. So this is the birth of SUGEN. And SUGEN stands for Schlessinger Ulrich Tech. S is Schlessinger, U is Ulrich.
Oh, it's named after you and your and Axel. Wow, that's interesting.
Yeah. So and then the other founders was someone called Stephen Evans-Reek, which was an Englishman, which was an investor. So we put together SUGEN, and he gave this name. So this was the first biotech company who was dedicated to developing drug for cancer. And you can see how this originates from my past, in which I start to work on EGF receptor, FGF receptor, and all those things. And we cloned many receptors. So FGF receptor is just one of the examples. So we started SUGEN, and this was quite a big success because we were among the first company that was doing that, and which led eventually to acquisition by some companies, and all the drugs of SUGEN have been developed eventually by Pfizer, because there were multiple acquisitions. And this benefited the shareholders, and also royalties payments for drugs and all that. So this was financially very, very good.
Mm-hmm. So I know SUGEN went public in 1994. Did you ever got the chance to go to New York and ring the bell at the stock exchange, if you know what I'm talking about?
So I was not an employee of the company. So in the company, I was a member of the board and consultant. I always stayed in my academic department, in my academic lab and department. And so I was involved in the roadshow for SUGEN getting to public. And it was received very nicely, and then of course it was sold, and eventually 2 cancer drugs came from that.
Mm-hmm.
So during the process, I realized into the value of structural biology for getting drug discovery. And then the last president of SUGEN was someone called Peter Hirth. And so Peter, and this was the beginning of a period in which there was interest in what they call structural genomics, solving structures in order to get knowledge and drug. So there was another scientist, a professor from Berkeley called Sun-Ho Kim, a Korean guy, former Korean guy, and we put together Plexxikon. So Plexxikon, the idea of Plexxikon was to use structural biology to develop drugs. And since, so in SUGEN, I was involved in every aspect, and there was a sort of agreement between SUGEN and my lab where insights have been licensed and patents have been licensed. But in Plexxikon, I decided that in order to be involved in the company, I sort of asked to be the chairman of the board. So in all of my other rest companies, I was chairman of the board. So I was chairman of the board of Plexxikon from the foundation until it was acquired by Daiichi Sankyo.
Yeah, and my question is, I know SUGEN went public, and I think back in the 90s, it was as big as Tularic. You probably heard of it. I've got the co-founder of Tularic was on the podcast. Why did you sell SUGEN? I'm just wondering.
Well, when in SUGEN, I was at this stage, I was still very naive in the business side, and once the investors had an opportunity for an exit to guarantee their returns, you cannot stop them. This is what they want to do. The same was true in Plexxikon. I would have voted to continue and develop these companies until they were independent and successful. This would be my preference, because I was not in a hurry. But this has to do with the spirit of the business, in which when investors have a guaranteed way for an exit where they will get return, they will take, because they will de-risk it, And if it's fivefold, tenfold, or even sixfold or less to what they invested, they're ready to do it. And at that stage of the company, I did not have any control. So it was not my decision.
Mm.
But I did not vote against, because there was no point to make a sort of a statement where I know that I would lose, because it was clear that that's what they want to do. They controlled 80% of the shares of the company.
I see. So was there any like AI involved in Plexxikon's platform for drug
Well,
discovery?
we are talking about here late 90s and
Oh, so there wasn't.
2000. 2000. We developed a screening method, and we used software to detect interaction between ligands, and then really refine. So it was a very high tech it was founded in 2000. So.
Are there
And
still any drugs on the market that are developed by Plexxikon now?
two of them.
See.
The first melanoma drug, BRAF inhibitor, and another drug. So I can send you all this information. So Plexxikon is responsible for developing two cancer drugs. And I can, by the way, send you my CV and all those things if you want to add it to information that you want to have.
Let's see.
So two drugs were developed with Plexxikon.
Yeah, so I have a question. It's not really related to you, but it's in the drug discovery industry. So recently, I think the Stanford's former chancellor, Mark Levine, he founded Zara, about the company that uses AI to generate molecules to treat disease. So it's like a drug discovery, but it uses AI. And I think it just got like $1 billion in funding, which is, I think, a record high for an early stage biotech startup. Do you think AI could push the biotech field in a whole new direction?
Well, look, at this stage, it's a promise, but it has to be fulfilled. It's clearly a powerful technology, but it's clearly also the raising of money has an element of a hype. So I think it's clearly a great development, and a lot of investors are betting that this will work in the future. We don't have yet any clear path for this to happen. So you describe what's the name of the company? I don't know this information.
So is Zara, XAIRA? It's like an AI biotech company, basically using AI to discover more drugs.
And it's
basically.
Mark Levine?
Yes, it's Mark Levine. I mean, you probably I mean, he was on the news recently.
I know Mark Levine very well. I know him from the time he was a postdoc at Columbia University. I know him from the time he was president of the Rockefeller University. I know him. He's a good guy. I know him very well.
Okay. Yeah, so next question is, you co-founded Coton Pharmaceuticals. So can you tell the audience what Coton does?
So Coton basically was so as we develop our science, we elucidated the mechanisms of action at molecular level.
Mm-hmm.
And then, of course, we solved the crystal structure of the extracellular domain of KIT, which is one of the receptor tyrosine kinase involved in different cancers and different biological responses. So we found a way how to block it through therapeutic antibodies. One of the things that I ignored, and it was my mistake, is the value of therapeutic antibodies, because my lab was the first to develop a therapeutic, an antibody against EGF receptor, and I did not put enough value into this for developing drugs. And then, of course, they're quite successful therapeutic antibodies. So Coltan was basically founded on the basis of using Achilles heels in receptors, which were discovered through structure analysis to develop new therapeutics. And Coltan, at a certain stage, merged with a company called Celldex, and Celldex is now developing this therapeutic antibody for treatment of different allergies. And Celldex is a public company, and you can see what they're doing. You have all the information. So this is an antibody that we developed, and ideas came from my lab. It's an antibody.
Are you still involved in Celldex? Or
I am on their scientific advisory board.
I see. Okay.
It's a public company.
So let me ask, I know you just had a new startup or recently new startup. It's called Opna Bio. It just raised $38 million. So unlike your previous companies you founded, what does Opna Bio focus on?
So I founded 2 companies. So all my first companies were based on my own ideas and Axel Ulrich ideas, some of them. But at certain stage, because of my success in companies, at certain stage, some people shared with me their ideas, and their ideas were very good, some of them better than mine. And so we discussed, and if it was scientifically interesting to me, I helped them set up a company. And I have set up 2 such companies. One of them is called Inozyme, which is also a public company. Okay, I founded, and I was a chairman of the board until it went public. And I'm still under SAB. So this is a company that is using enzyme replacement therapy to cure babies which have deficiency in an enzyme which causes severe calcification, and they die very early. And this company is public, and the drug is now in phase 1/2. We have generated this enzyme, and you can find everything because it's a public company. Okay, a second company which I founded, and I'm also chairman of the board, is called Opna Bio. The scientific idea is not mine. It was someone called Doug Hanahan, who is an American, and he's basically a professor at EPFL Institute at Lausanne. He found out that a gene called FMRP, which is normally involved in retardation, a loss of function in this gene cause some kind of genetic development and retardation. Somehow, this gene is co-opted into multiple solid tumors, and then it causes a switch off to the immune response against these tumors.
I see.
And and you can there's a very nice website. You can look Opna Bio website describing all what we can do. Another thing is the company licensed since basically Plexxikon became part of Daiichi Sankyo. Daiichi Sankyo decided to move all her assets into antibody drug conjugates. So they basically licensed to Opna Bio all their assets for small inhibitors. So we have now drugs which are in clinical trial, which are small inhibitor. You can see all these drugs described in the website of Opna Bio. Okay, so this group, Inozyme, is a public company. You have a nice website. I am not involved anymore on the board, but I am on the SAB. Opna Bio, I'm chairman of the board and member of the SAB. It's not a public company. It's a company that develops drugs for cancers, and FMRP is the key program in that company. You can see all the details, and if you need more information, I'll send it to you as well.
I mean, I know you mentioned that Opna acquired a portfolio of patents from Plexxikon. You mentioned how hard were these negotiations? Like, was it hard to ask Daiichi to?
Well, actually, so this was quite easy for the following reason. So Daiichi, I was founder of Plexxikon, and their president was someone called Gidon Bolag, which was the president of of the Plexxikon, and then at certain stage, he decided to move and be the president of Opna Bio. So the person who was running the program in Daiichi Sankyo moved to, so they were very trustworthy. So you see what I'm saying? So it was like a basically friendly transfer of assets from one company to another company when I was one of the founders of both companies.
Mm-hmm.
So it was not a difficult negotiation because we all knew each other, and there was an interest of continuing to do because they changed their scope.
Mm-hmm. So, I mean, question is, I mean, you founded so many companies. You mentioned some of them are still in the stock market, some of them were bought by other larger pharmaceuticals, and some are still private. So, what do you learn from forming all of these companies and just kind of watch them grow?
Yeah, well, it's a real pleasure to see that. It's a real pleasure to come one day with an idea and then talk to colleagues and see that this is a good idea, and then talk to investor, raise money, and then eventually hundreds of people are treated and you see an effect. So this is really very satisfying. Okay. And I have to tell you that I never made any strategic plan to do that. I'm doing all that because this is all enjoyable and fun. I don't have to work. I'm at a retirement age and I have enough income not to do anything, just enjoy life. But I consider that most enjoyable, to do what I really like as a scientist. And so the market, the public market and the institutional investors are subject to a lot of different inputs, and so there are from time to time situations where there are hypes, and then the AI is certainly an important thing, but to some extent the response is clearly they pick up everything without much of selectivity because the returns are not going to be quick.
Mm-hmm. So, I mean, just right now at the biotech industry, what companies right now do you think could have a similar impact to the biotech, like Biogen and also Genentech?
Well, I have not been in. I mean, what are the current new companies which are?
Yeah, like do you think could have the similar impact to the biotech industry, like Genentech and Biogen?
Well, another company which had a tremendous impact is Regeneron. And Regeneron is an independent company, so the early companies, the rate of success in startups into developing drugs and succeeding as a business is very low. It's very, very low. And you don't know about many of them which disappeared. So it's difficult to know which of them. If I would know any one of them, which I think is great, I would probably invest my own money into it. But I'm investing my own money into much more safer in order to keep it for the next generation.
Mm-hmm. So I see that you're part of Yale Ventures. So is Yale Ventures like the venture capitalist firm, or what is it? What role do you play in Yale Ventures?
I don't understand the question. Say.
So I see that you're part of Yale Ventures, like you have a
Yeah. Yale Ventures. Okay, so Yale Venture is basically a name for the office that that manages the intellectual property and the business in the business area in every topic. But the largest portfolio of Yale Venture is the life science, but they do other things too. So all faculty members of Yale are associated, if they want, with Yale Ventures, but there's nothing there beyond myself being just another member of the faculty.
I see. So what advice did you receive during your career that shaped your professional development or success?
Very, very simple advice. You have to like what you're doing. I was I always looked for having intellectual pleasure and enjoying the science and looking forward for another day of going to the lab. That was my advice. You should really, I never did anything for, I optimized it in order to get nice income, but it was never the motivation. And I always realized here it's a great opportunity. I want to find some new discoveries and go with the science when we can develop some drugs.
Mm-hmm. So, is there any other advice you would give to students who want to become scientists or entrepreneurs, like specifically?
Well, this is the same advice I give them. Throughout my career, I always had fun. Not that I did not have setbacks or failures. Of course, everyone has. But I always had fun. Science is, there's so much to do in science. And there's so many questions to ask. So it's really, I feel always like a boy that comes to a toy store, and has to choose. So this is my advice. If you don't have fun, don't do this.
Mm-hmm. Thank you, Dr. Schlessinger, for being part of this episode, giving me the opportunity to talk to you so people can get to know more about your insights in tyrosine kinase and also entrepreneurship.
Okay, thank you very much.